Bismillahirrahmanirrahim. So we are looking at chapter 18, Biotechnology and Genetic Modification. There's a bit of repetition in this chapter, so probably because obviously so that the concept becomes clear after reading it twice, thrice. Right now, for explanation's sake, I won't be going into the repetition. So we'll cover the basics that we need to know, and it's mostly reading, honestly. So there are two terms that you need to be familiar with. One is biotechnology, and it states over here it is the application of biological organisms, systems meaning biological systems, or processes, that is biological processes, to manufacturing and service industries. Okay, so basically using biology to the benefit of human beings. And genetic modification, and this definition will come again formally later, but it's the same as given over here. Genetic modification involves the transfer of genes from one organism to usually an unrelated species. Right now, species meaning from one bacteria to another, as you'll see, from humans to bacteria. That is what's normally applied. It could be from bacteria to plants. We'll see that as well. Viruses can also be used in this case. So all of that will come in later.
Biotechnology is used in multiple industries and all, no?
Yes.
Okay.
So an example of biotechnology is the use of enzymes in washing powders. So they mention it
Okay.
later, but since you asked, right? So when you have food stains or any sort of stain that is based biologically, chemically, it contains proteins or fats, lipids, right? And so that is does not come off easily just by regular soap or just like washing it with water. And so washing powders contain enzymes, right? Lipases and proteases, and those digest the protein and lipid, whatever it may be, and breaks it down into smaller molecules. And then the smaller molecules are easier to get rid of from between the weaving of the clothes, right? So that is an application of, as an example of biotechnology where enzyme activity is employed for our benefit. There are many other examples. So, right, bacteria are widely used and they are genetically modified for different uses. So what happens is, if you remember the structure of bacteria, they don't have a nucleus. They have one main circular DNA, and then they have smaller circular DNAs, pieces of DNA, which are called plasmids. And those plasmids are very important. So what can be done is external DNA can be incorporated, can be added to those plasmids, and then those plasmids are put into the bacteria. And because the DNA sequence, coding sequence is universal, right? So if a triplet code uh, is for a certain amino acid, it's the same whether it's in plants and animals, human beings, bacteria, viruses, whatever it is. It's the same code. And so an example is human insulin. That's come in earlier as well. It's going to come over here. And so human insulin is mass produced using bacteria. So the gene that codes for insulin in human beings is taken, it's introduced into a plasmid, and that's put into a bacteria. And so you've got genetically modified bacteria. And those bacteria are then grown where they produce human insulin proteins, and then that is extracted and right then obviously used as medicine for people who are suffering from diabetes. You've got Other examples, a very basic example, and nothing fancy. Like for example, making bread. So that's using yeast to, undergo anaerobic respiration, where it reduces carbon dioxide in the flour, and that causes the bread to rise. Right. So that is basically biotechnology. Making wine, again using the anaerobic respiration and fermentation processes. Right. uh, Cheese as well, right? Making yogurt and cheese, and that involves bacteria and fungi and yeasts, depending on what's being done, right? Antibiotics, right? The OG antibiotic, penicillin, right? The first antibiotic ever discovered, uh, is produced by fungi, by some bacteria as well, I guess, as you mentioned over here, uh, right?
But
And
antibiotics are used to treat bacterial infections, na? And
yes,
they are also made by bacteria.
they could be made by bacteria, they could be synthetic, and they could be obtained from other sources as well, right? The purpose is that they
What is
are able to act on bacteria and somehow stop their growth or kill them off, or both.
what does synthetic means?
Synthetic means that it's not found in nature, and it was devised, like the chemical was designed by human beings entirely.
Okay.
So it's not something that you'll be able to find in nature. Now, one question is, why are we able to uh, get antibiotics from nature, and even like from other bacteria, right? Why would one bacteria produce a chemical that would kill a different bacteria? Any ideas?
So can you repeat the question again?
Why would a fungus or a bacteria produce a chemical that is able to kill other bacteria?
Because they can overcome, like, because they have the same characteristics. That's
Okay, so right, just like we have ecosystems that obviously involve us, but even if we take ourselves out of the equation, we have ecosystems where you've got plants and insects and birds and, you know, all these other animals that are interacting with each other, and you have this food chain and food web and all of this. That's also happening in the microscopic world, right? So it's not like all the microorganisms are having a huge party and living a very, jolly life together. So there are predators, there are so to speak, carnivorous microorganisms. There are the, uh, ones that eat others. There are ones that eat, consume minerals or decomposers or others. So there are all sorts of microorganisms, and so, right, they attack one another as well. And so antibiotics are basically defenses, chemical defenses produced by some microorganisms against others. And so, right, we are able to use that to our benefit as well. Okay, then is sewage disposal. So this comes in later as well. So this is just all of this is sort of like a summarized introduction of things that they will mention later. But right, if you just get an idea of what's happening, we won't have to linger around when it does show up in the text. So sewage disposal, again, we are using bacteria that are decomposers to, know, utilize the nutrients in sewage and break down organic matter in that sewage, so that the water purifies, and then you use other chemicals and filtration, sand and everything to clear it out. Okay, so right, biotechnology does not only concern microorganisms, cell cultures and enzymes, as I mentioned, are also used in modern developments. So where you got fermentation, right, involves the anaerobic respiration of yeast converting glucose to ethanol, right? So that's that. Over here, again, just reading, it's talking about how ethanol can be used as a source of fuel, but the problem is that the sugar cane that needs to be grown is expensive, and therefore the fuel is expensive. And which country is, producing ethanol from sugar cane or from soybeans and oil palms, right? As those alternative fuel sources, right? And then obviously the problem of human interaction with the environment. So we, when we are growing these forests, right, in fact, not forests are being destroyed, not growing forests, but forests are being destroyed to plant soybeans and oil palms, and which are affecting habitats such as for orangutans, right? And they have now become endangered, right? They're on the verge of extinction. Bread, as I mentioned, right? So again, anaerobic respiration, and carbon dioxide is produced, which causes the flour to rise, as you can see in this picture. The dough at the back has risen, and any ethanol produced and carbon dioxide, when you put that into the oven, the ethanol evaporates, the carbon dioxide also escapes, and that's why you have got these sort of bubble like spaces in bread, because that's where the carbon dioxide was. Fruit juice production, right? So pectinase are enzymes that are used.
Like,
Hmm?
so like ethanols are not found in nature,
They are.
but like they are produced from like fermented sugar, no?
They're not produced from fermented sugar. They are produced because of anaerobic respiration of yeast. I mean, that's one thing, right? I mean, other like organisms might be able to produce ethanol as well, right? We're only talking about yeast in this case. So when yeast has sugar as a fuel source, carbohydrate as a fuel source, but it's put in anaerobic conditions, so oxygen is lacking, and so when it does respire, when it does consume that carbohydrate, it will convert it to ethanol.
Yes.
Okay, and that ethanol again is used in fermentation to make wine and all of that. It's used in, right, the ethanol is not used in bread. The process of anaerobic respiration is used in making bread to make the dough rise, right? So in that case, the ethanol is not the important product. The important product is the carbon dioxide, which is going to increase the volume of the dough, right? Wine making is going to come after fermenters, so antibiotics, and could have been here somewhere. Anyway, yeah, so here we are, right? No, this is for fuel. Anyway, point is, right? Uh, so that is one application. Now, fruit juice production. Pectinases or pectinase is an enzyme that's used to make fruit juice from fruits. And what this does is it's taken from a fungus, and they've given the name over here, Aspergillus niger, and it breaks down the carbohydrate pectin. And that pectin is a carbohydrate. It is a substance that's present between cell walls of different cells to sort of like stick them together, right? So it's pectinase digests pectin, and so that polysaccharide is broken down into, right, smaller molecules. And what that does is, it, because it is a carbohydrate, it makes fruit juice sweeter than you would expect fruit to be. So, right, I mean, apart from the added sugars that are in juices, right, apart from that, because there's more sugar content. Remember that mono and disaccharides are called sugars. Polysaccharides are not called sugars because they're not sweet and they're not soluble, etc., etc. If you recall the chapter on biological molecules. So pectin is a polysaccharide. It's not a sugar, but when it's processed using pectinase, it is broken down and it becomes sugars after digestion with the enzyme, right? So that's why fruit juice becomes sweeter than the fruit itself was. Also, right, the enzyme makes the fruit juice transparent, right? So it clears up all the material that is sort of suspended in the fruit juice and it makes it clear. So that's double benefit. It makes it sweeter and also makes it clear. Biological washing powders, I already mentioned that, right? So they're mentioning grease and oil and protein, right? Blood and egg and all of that. And so enzymes and biological washing powders are useful. Uh, right? It also what they're also useful because now you don't need to use hot water to get the stains off. Uh, if you do use hot water with biological washing powder, then you'll end up denaturing the enzymes, and so they won't work. So, right, it makes life easier. You also have to be careful about the temperature of water that you're using. Then fermenters. So fermenters are these huge, right, industrial setups where they have many different applications. So, right, producing penicillin, producing alcohol, right, wine, and all that, producing insulin, uh, and right, making lactose-free milk. So many different applications of fermenters, right? Lactose-free milk. So lactose is right. Some people cannot digest lactose, and so it remains undigested, causes diarrhea and stomach pain and gas, and it is unpleasant. And so lactase enzyme is used to digest the lactose to glucose and galactose. Lactose is a disaccharide and it's made from glucose and galactose. So the lactase enzyme is added to these fermenters, added to milk to break down the lactase, and right, so that way the people who are lactose intolerant can consume the milk and it's possibly sweeter. I've never tried it honestly, but again, right, so you have more sort of sugar in it, so to speak, as compared to regular milk, right? Another benefit over here is, right, the lactase that's used is in the form of immobilized lactase on the surface of beads, right? And so the milk is passed over the beads so that the lactose sugar is, digested. And so this way, the enzyme molecule is not sort of, utilized. It can be used for many different batches because, as you know, enzymes are not used. It's a catalyst, so it does not take part in the reaction. It's not used up. But if you add like liquid enzyme to the milk, and it becomes mixed in with the milk, even if it's not being, chemically used up, you can't extract it back out of the milk. So using immobilized beads, right, immobilized enzyme on beads keeps the enzyme in one place, whereas the milk is flowing over it and being, the lactose is being digested by the lactase enzyme, and so that makes it reusable, right? The enzyme. Any questions so far?
No.
Okay, then you got the production of antibiotics. Alright, so when microorganisms are grown in fermenters to produce antibiotics, it is not their fermentation products that are needed, but complex organic compounds called antibiotics that they synthesize. Right? Penicillin. So they're talking about fermenting tanks that can go up till 100,000 liters in capacity, and they're filled with nutrient solution. And sugar is, right, carbohydrate source is sugar, mainly lactose or corn steep liquor. Uh, right, and they've got some amino acids as well over there. Mineral salts, the pH is between 5 and 6, the temperature is about 26 degrees. It is aerated, right, and stirred to mix the air, so it's not, right, it's not anaerobic, what happens is that the culture of the appropriate microorganism that's going to produce the penicillin is added to the nutrient liquid and allowed to grow for about 24 to 48 hours, which is 1 to 2 days. Sterile conditions are essential. You don't want any other microorganism growing over there because it's going to spoil. Right? They're going to grow and they're going to make their own products, and so your penicillin is going to be contaminated. Uh, this is the important part. As the nutrient supply reduces, the microorganisms begin to secrete the antibiotics into the nutrient. Right? So they need, right, uh, low nutrient levels to actually produce the antibiotic. Now here's the question: if they need low nutrient levels for antibiotic production, why do we initially add all those nutrients to begin with? We can start with low nutrient conditions and not have to wait for 24 or 48 hours and get penicillin from the start.
Uh, can you please repeat that question again?
The textbook is telling us that the microorganism secretes the antibiotic, in this case penicillin, when the nutrient level is low. So the question is, why are we adding all these nutrients in the beginning? Why don't we start with low nutrient levels so that the microorganisms produce penicillin from the start?
Like they get absorbed the nutrients.
Why?
Because like in the fermented they get absorbed the nutrients and then later on they are used.
Okay, you see over here, a culture of microorganisms is added to the nutrient liquid and is allowed to grow for 24 to 48 hours. We add the nutrients so that the number of microorganisms increases, right? So the more microorganism there is in the fermenter, when the nutrient level drops, it will you will be able to get more penicillin out of them. If you've got less microorganisms, then doesn't matter how big the fermenter is, right? You're going to get lesser penicillin because you only had a few microorganisms, right? And so that's why you add the nutrients to let the microorganisms first grow and then produce the penicillin when the nutrient level drops.
Okay.
Right, insulin is something they'll mention again later. So I did say that there's quite a bit of repetition of concepts over here. Uh, so insulin is coming in later. Mycoprotein is a protein-rich substance. It's basically extracted from fungi, and its texture is like meat. So it's used as a meat substitute. It contains low amounts of saturated fats than actual meat. It has zero cholesterol, so it's supposedly healthy. I say supposedly because, right, I've never had it to eat, uh, and it does not contain any animal products, so it's vegan. So that's good as well, right? And you can add flavors to it and make it like however you want it to be. Its manufacture has been developed so it can be made commercially. It is fermented in a similar way to antibiotics. So we right whatever they mentioned in the production of penicillin, and enzymes using glucose. Right, sorry, it is fermented in a similar way to antibiotics and enzymes using glucose and salt as the feedstock. Okay, over this table gives all the conditions that you need in a commercial fermenter. Right? In this case, in the manufacture of an antibiotic. So the temperature, the right pH, oxygen, nutrient, waste products, and all that. In the case of antibiotics, right, the nutrient fluid containing the antibiotic is filtered, and then the antibiotic is extracted by crystallization or other methods. Any questions?
No.
All right, then we come to genetic modification. As I mentioned, the right definition
Um,
is given
by
here.
the way,
Yes.
can you show the visual representation of how the fermenters work?
So they have an illustration, I believe, over here. So this is how it works, right? You have the main, right, nutrient stock. You're putting in more feed stock. You've got the microorganism in here. You're pumping in air through it. The paddles stir the liquid to evenly distribute the feedstock and the microorganisms and the air, right? And then whatever they're producing is taken and filtered, right? And the waste products are removed, dark blue over here in the pipe. And the filtrate is what contains the antibiotic in this case, or could be enzyme or mycoprotein, depends. And then it's extracted and crystallized and processed, however, right? So this is the industrial fermenter. This is a smaller scale laboratory fermenter in the picture over here, right? And these are large scale, okay, in industry. All right. All right, genetic modification. So once again, genetic modification is changing the genetic material of organisms by removing, changing, or inserting individual genes, right? Those genes are usually coming from a different species, right? A different organism. So, right, you can remove something, you can change something one for the other, or you could insert something new. Right, the process often uses bacteria because of their ability to make complex molecules and their rapid reproduction rate. So they can, right, sort of the fastest reproduction rate in bacteria is like once every 20 minutes, so 3 times per hour, which is fast, right? And so within a matter of a few hours, a day or two, you're going to have a huge column of bacteria, right, producing whatever it is that you want. So how is the genetic modification of bacteria, right, done, and what are they used for? So bacteria are microscopic single-celled organisms with cytoplasm, cell membranes, and cell walls, but without a proper nucleus. The genetic control in bacterium is carried out by a piece of circular DNA. Right, there are also other smaller circular DNA called plasmids, and right, that circular DNA is cut. So basically, plasmids and foreign genes, right? So from other organisms can be included in that. And so one example of that is obviously human insulin, right? So when human DNA is added to a suspension of plasmids, some of the human DNA attaches to some of the plasmids. The plasmids are first cut so that they're no longer circular and they're sort of open. And then the human gene is put in, it attaches and it closes again, so it becomes a circle again, right? And then they are treated so that they close up. The bacteria are stimulated to take up the plasmids, so the bacteria reabsorb the plasmids, right? Given suitable nutrient solutions, these bacteria containing the plasmids reproduce rapidly by mitosis to make millions of cells. Each daughter bacterium will contain the same DNA as the parent, and so that DNA that you introduced initially is going to be present in all the cells that replicate, right? And so all of them are going to produce the protein that is coded for the by the gene that you introduced, right? An example again being insulin. And they're also, this is also done in, fermenters, and the product is extracted and purified and all of that, right? What we just said. So over here is the process. So you've got this bacteria, these green circle are plasmids. Those plasmids are cut, right? And then these red lines are, for example, human DNA. And so those are put in and they join up, right? And then they're put back into the bacteria. So that is basically like the basics of genetic modification of bacteria, right? Or genetic modification in general. Any questions?
No.
Okay. So what are they saying over here? The plasmids are said to be vectors. They carry the human DNA into the bacteria. The technique is sometimes called gene splicing. So that's something you may want to know, right? Another, like, what's the technique of introducing human DNA into bacteria? That's called, so the name is gene splicing. Now, using plasmids is only one type of genetic modification, right? Viruses can be used. DNA can be inserted directly without a vector. The donor DNA can possibly be synthesized from nucleotides rather than taking them from cells. Yeast may be used instead of bacteria, right? So there are many different ways this can be done. The outcome is always the same. DNA from one species is inserted into a different species and made to produce the protein that it codes for, right? The normal proteins. So again, they're talking about human insulin, right? There are also bacteria or viruses that are used as vectors to deliver genes. For example, a bacterium is used to deliver a gene for herbicide resistance into crop plants. So that's again coming up in the following pages. And right, so applications of genetic modification. Again, they're mentioning insulin over and over again. GM crops, like we just mentioned. So human insulin, again, right, taking human insulin gene, putting it in bacteria, growing it in fermenters, causing it to produce insulin, right? And the good thing is that earlier insulin used to be taken from animals, and so that used to have reactions and rejection in the human body. And it wasn't as efficient because obviously it was somewhat different from human insulin, so it didn't work as well. But now you are synthetically producing human insulin, and so it's going to work exactly as actual human insulin does, and there's no rejection, there's no reaction, it's all good, right? And there are no ethical concerns about using animals, there's no religious concerns either, right? So very good. Then you have GM crops. So the crops can be modified to become resistant to insects, to herbicides, to other conditions, right? For example, pest resistance. So there's a bacteria called Bacillus thuringiensis. It produces a toxin that kills caterpillars and other insect larvae, right? And so that is used, right? It is a toxin that has been successfully introduced into some species
Sir,
of, yeah.
what is herbicides?
Herbicide is a chemical that uh, kills plants,
Okay.
right? Just like, the bug spray that you use is technically it's a pesticide,
Yeah.
right? So, right, this bacteria that produces a toxin for these caterpillar and their larvae, they're introduced into crop plants such as maize, right? Because the maize has these stem borers that eat through the plant, and so the plant does not stay up straight, it bends and drops, and so that kills the plant. And so what they do, or what they've done, is that they've incorporated that toxin gene into the plant, and so it's naturally producing that toxin that will kill these pests. So when the pest comes in, it starts eating the plant, the toxin is in the plant, and they'll die. And the gene is also passed on to the plant's offspring. Unfortunately, there are signs that insects are developing immunity to the toxin. Okay. Then you've got herbicide resistance, right? So there's a herbicide called glyphosate. It will kill any green plant, right? And so some crops have been introduced with a gene which breaks down the glyphosate in the cells, right? And so this makes it resistance. This gives it resistance to the herbicide. And so what happens is when, right, you've got these GM crops with the herbicide resistance, when you spray glyphosate in the field, it will kill all the weeds and other plants that you don't want. But this plant, because it is resistant, it can break down the herbicide into harmless products. Then, right, it will survive. Now the problem is that some scientists suspect that glyphosate is carcinogenic and it is being banned from use in some countries, right? So obviously there are issues with all of this, right? It's not a picture perfect world. Now if I just push this a little bit, we'll be able to complete it in this session, and that would be very good for both of us. Yeah, so just stick with me. All right, providing additional vitamins. So there's a variety of crop that has been uh, made, and it's used in, I think, Bangladesh and somewhere else, not in all over the world, where it has extra vitamin A. How does it have extra vitamin A? A gene for beta carotene, which is a precursor. Precursor means this beta carotene is processed and converted into vitamin A, right? So it's not directly vitamin A, it's beta carotene, right? It can, so that gene can be inserted into these plants, right? And so when we eat that, our body processes beta carotene into vitamin A. And so in regions where vitamin A deficiency is prevalent, right, which often leads to blindness, eating these crops with additional vitamin A supplements, right, is going to be helpful. So then we talk about advantages and disadvantages of genetic modification. So in terms of crops, right, one objection is that only the farmers and the chemical companies in the industrialized world have benefited, right? So the developing world, the so-called third world countries, right, they don't have access to this. So that's a problem, except for China, right? And many trials are being done. On a side note, I would say, right, a lot of this is, the advertising material, right? Reality is not how they present it. I'm just saying that on the side so you can research and get to know what's really happening. Okay, as far as the syllabus is concerned, obviously they're going to ask you what they've given in the text, and that's what you need to know. But just so that you know, right, this is just advertising. Okay, so inadequate intake of iron is one major, is one of the major deficiencies in the world. And so some plants, like an enzyme in some plants, plant roots enables them to extract more iron from the soil. And so the gene for this enzyme can be transferred to plants like rice, having them contain more iron. Some acid soils contain levels of aluminum that reduce yields, right? So a gene introduced into maize produces citrate, which binds to aluminum in the soil and releases phosphate ions. After 15 years of trial, the GM maize was made available to farmers, but pressure from environmental groups has blocked its adoption. So again, right, there's some problem because of which they weren't able to do it. It's only used in the USA, Brazil, and Argentina. As a result of irrigation, many agricultural lands become salty and unproductive. So introducing a gene for salt tolerance, right, can obviously maybe make it possible to grow plants in these regions. Then you've got the gene for nitrogen fixation from leguminous plants. And so those could be introduced to cereal crops, however, like for example, which can, fix nitrogen, even though that's not happening naturally. But if you introduce it, they might be able to fix nitrogen and have higher yields, right? More protein, right? Higher yield. And so that would reduce the need for fertilizers. Genes for drought resistance would make dry areas available for growing crops. So there are, right, obviously concerns, ethical concerns, and right, there are concerns that these genetically modified plants might crossbreed with wild plants or weeds that you normally kill off using herbicides, and these genes might transfer to them, and so they might become resistant to weather conditions, they might become resistant to pests, they might become resistant to herbicides, and so they become super weeds. Also, we don't really know what the effect of these genetic modifications are on human beings. Even though in the book they're saying that, oh, these plants are only introduced after years of testing. But again, right, this is, as I mentioned, advertising. But as far as the syllabus is concerned, right, that's what they're telling. So in conventional crossbreeding, the genes transferred come from the same or closely related species. However, in crossbreeding, the whole range of genes is transferred, and this has sometimes had bad results when genes other than the target genes have combined to produce harmful products. Genetic modification offers the advantage of transferring only the genes that we want, that are required. The differences between the genetic makeup of different organisms is not so great, right? And not all genetic modification involves the transfer of alien genes. In some cases, it is the plant's own genes that are modified to improve its success. I mentioned the superweed problem, so that's over here. To prevent the transfer of pollen from GM plants, other genes can be introduced. These genes would stop the plant from producing pollen, so they will not be able to naturally reproduce. Okay. And in the last part of the chapter, GM food. So again, the golden rice is the rice that has beta carotene gene for added vitamin A and all of that. So GM food is prepared from GM crops and they're aimed at increasing yields rather than changing the quality of the food. It is possible to improve the protein, mineral, or vitamin content of foods and the shelf life, right? So it doesn't spoil as quickly. One of the worries is that the bacteria for delivering recombinant DNA contains genes for antibiotic resistance, right? And so why does it contain genes for antibiotic resistance? That is the method used to select the bacteria that have taken up the gene that we introduced. So not all the bacteria take up the gene that we put in, and so to distinguish them, we introduce antibiotic resistance into the bacteria, and then we grow them and we add antibiotics. So the ones that die off means that they did not have the gene we put in. The ones that survive means that they do have the gene that we put in, and then we grow them and we use them. So the problem is that they do have antibiotic resistance. So if they are able to sort of pass that antibiotic resistance to other microorganisms, then you're going to have antibiotic resistance problems, right? So that's a major concern. They say that although there is no evidence to suggest that this happens in experimental animals, the main biotech companies are trying to find methods of selecting bacterial vectors without using antibiotics, and at A level you will learn some of those methods. Another concern is that GM food could contain pesticide residues or substances that cause allergies. This does happen, right? Even though they're saying that they're rigorously tested for toxins and all of that over many years, but it does happen. The GM products must be passed by a series of regulatory and advisory bodies, and we know how that works, right? How rigorous they really are. And right before they're released onto the market, in fact, only a handful of GM foods are available. One of these is soya, which is included in one form or the other in 60% of processed foods. Golden rice, right? Again, beta carotene, all of that. Some argue that there's a danger of the precursor of this beta carotene, right? Like not precursor of beta carotene, but the beta carotene itself changing into other toxic chemicals once eaten. And that's why Philippines and Bangladesh are the only countries where this is used at the moment. Right? So golden rice is called golden rice because it's actually yellow, as you can see over here. The white one is the regular rice. There are also concerns about the reduction in biodiversity, right? So if these genetically modified crops are able to survive better than the wild crops will die off, and so you'll only have genetically modified crops, and that's going to reduce biodiversity, right? So subsistence farmers could also be tied to large agricultural suppliers who may manipulate seed prices. Why? Because these are crops are modified to not reproduce on their own. They don't produce pollen, or the pollen is not fertile, right? And so every time the farmer has to buy seeds from the company. Apart from specific hazards, there is also a sense of unease about introducing genes from one species into a totally different species. Right? This is something that does not happen in nature, and so the long-term effects are not known. At least at least some of the protests against GM crops may be ill-judged. Okay, finally, bacteria. So again, right, they're mentioning the same thing. Bacteria are useful in genetic modification, rapid reproduction, uh, lack of like there are no ethical concerns about manipulating bacteria. The genetic code is shared between all organisms, and so they can produce whatever the protein for whatever genes you're putting in them, whether it's an enzyme or a hormone or anything else. There are some concerns about the use of genetically modified bacteria, such as to produce human insulin, because there is the risk that the bacteria could pass the human gene they carry to other bacteria. Now, what's the problem if some other bacteria starts producing insulin? We don't know. And so that is the risk, right? Unknown consequences. And with that, we have covered the chapter. Any questions?
No.
All right, so it's pretty straightforward. The underlying concept is very simple, and they've basically just gone over a lot of other examples of where genetic modification biotechnology are implemented, right? And yeah, so this was basically just a lot of detail. So with whatever time we have left, next time we'll do classification, and then chapter 19, which is relationship of organisms with one another and the environment. If we have time left, then we'll do chapter 16 and 17 if possible, right? Otherwise, right, that's how far we go. Okay, and obviously we won't be able to do past papers. Now, that being said, let me just be clear that my student is not an appearing student, and so, right, the syllabus 100% syllabus coverage and past paper requirement, like practice, is not required, right? It would have been good if we were able to do that, but right, we won't be, and that's okay as well. All right, so I'll see you next time, inshallah.
Okay. Allah.
Allah Hafiz.